Good day, this is Professor Will, CBMD PhD, and today's Monday, January 17th, 2022. It is 4:56 and 6 seconds a.m. Eastern Standard Time. Today's research, metabolic disease. So let's hop to it here. Discovery can hold the key to alleviating metabolic disease. A newly characterized fat protein could provide a missing link to explain a rare metabolic disease, while offering fresh insight into common disorders such as obesity and type 2 diabetes. Date January 13, 2022. Source: University of Edinburgh. Familial partial lipodystrophy type 2 is a rare disorder causing diabetes, loss of fat in the arms and legs, and overdevelopment of muscles. Yet the faulty protein, lamin A, is found in all cells. The researchers found that mice lacking the protein, TMEM128, which is mostly found in fat cells, have similar symptoms as familial partial lipodystrophy 2. The TMEM128 belongs to a group of proteins known as NETs that help ensure the cell's genetic material found inside the cell's command center, the nucleus, is correctly organized and read. They found that TMEM128 plays a key role in normal development of fat tissue and healthy metabolism by promoting expression of fat genes and shutting off muscle genes in fat tissue. And these effects appear to be due to TMEM128's ability to release parts of the genome that control fat metabolism away from the edge of the nucleus while recruiting muscle genes to the edge. And when the genes are at the edge of the nucleus, they tend to be shut off, and the scientists found that the mispositioning of genes in the mice lacking TMEM128 also occur in human patients with familial partial lipodystrophy 2. The study authors propose that because TMEM128 is found mostly in fat tissue, that it mediates the fat specificity of the defect caused by the faulty lamin A protein, previously linked to familial partial lipodystrophy 2. The studies on genetic defects that cause metabolic disease indicate they are complex, with many genes implicated, thus TMEM128, by affecting positioning of many genes, can similarly be involved. The findings could have wider implications for other metabolic diseases, such as diabetes, insulin resistance, glucose intolerance, and obesity, as well as muscle disorders and bodybuilding. The symptoms only become apparent in mice that were fed a high-calorie diet, consistent with the symptoms with familial partial lipodystrophy 2, which often appear later in life and require a carefully controlled diet. And this mechanism can explain why some diseases, such as diabetes, are only revealed when the body is under pressure, such as a high-calorie diet fed to the mice lacking the TMEM128 gene. The functional defects and other NET proteins are linked to many human diseases, such as muscular dystrophies, cardiomyopathy, blood and bone disorders, cancers, and premature aging syndromes. Other complex diseases could have a similar mechanism in which NETs affecting gene positioning cause small changes in expression of multiple genes so that the pathways still function, but at reduced capacity, thus producing symptoms only under certain circumstances. The part that is most interesting for researchers is the discovery of new mechanism for muscle growth observed in lipodystrophy that opens new potential avenues for increasing muscle mass that is lost, for example, in astronauts during space travel, in muscular dystrophy, or in muscle loss caused by injuries. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.